Welcome, everybody. It is really my pleasure to welcome you all to the annual Lurie Cancer Center Chung Lee Lecture. Dr. Lee actually also joined us. Professor Lee does not need introduction. I'm sure all know him. He has had tremendous career accomplishments and legacy. He joined the faculty of Northwestern University in 1974 as an assistant professor and rose through the ranks to professor of urology, and in 1992 was named the Jack Grayhack Professor of Urology. He has had more than 40 years of research experience in prostate cancer with a focus on prostate cancer biology, prostate cancer cell differentiation, growth factor signaling, apoptosis, gene therapy, amongst a long list of research areas. His research was continuously funded by the NIH and the DoD. And of note, he is the founding director of the prostate cancer SPORE at Northwestern University starting in 2001. And he was the PI till 2013 and has guided the successful development of many clinical programs, including clinical trials research. He is the author of more than 200 papers on benign and prostate cancer. He has mentored many graduate students and postdoctoral fellows. And I'm going to mention some names, and by no means that's a complete list. We have Isaac Kim, Erwin Park, Norm Smith, Scott Egner, amongst others. Dr. Lee is currently a professor emeritus at Northwestern University. This annual lecture was started in his honor several years ago. And I know Dr. Lee is here, and we normally would clap, Chung, but unfortunately now with the Zoom business, we're going to skip the clapping. I'm now going to move on, and it's my pleasure to welcome Dr. Kathy Kelly as our invited 2020 Chung Lee lectureship speaker. Dr. Kelly obtained her PhD from the University of California, Irvine, and completed postdoctoral training in genetics at Harvard University in the lab of Dr. Philip Letter. She joined the National Cancer Institute, starting out in the immunology branch, Division of Cancer Biology and Diagnosis, and then the Laboratory of Pathology at the NCI, where she rose through the rank and became the chairperson, and then branch chief from 2001 to 2013. In 2013, Dr. Kelly was promoted to chief the Laboratory of GU Cancer Pathogenesis. And in 2017, she also became deputy director for basic research at the Center for Cancer Research at the NCI. Dr. Kelly's lab and expertise, and she has done tremendous amount of work. I'm just going to briefly summarize some of the work that Dr. Kelly has done. Her lab and research is focused on the biology of prostate cancer stem cells, castrate-resistant prostate cancer, and PDX and organoid models of advanced prostate cancer. Early in her career, she established the identity and more broadly the functional categories of mitogen-induced early response genes. She later began focusing on the biology of aggressive prostate cancer, which actually I must admit is near and dear to my heart, myself on the clinical front, with an emphasis on metastases, cancer stem cells, and lineage plasticity with the objective of getting mechanistic insights into the specific genomic and phenotypic defined classes of advanced prostate cancer. We are delighted and thrilled to welcome Dr. Kelly as the 8th Chung Lee lecturer to honor the contribution of Professor Chung Lee. Her lecture is focusing on modeling mechanisms of therapeutic response and resistance using advanced prostate cancer patient-derived organoids. Kathy?
Yeah, thank you very much. I wish I could be there, meet you all in person, but nevertheless, it is really a distinct honor to give this lecture named for Dr. Chung Lee as a tribute to his many contributions in prostate research and his leadership of the prostate program at the Lurie Cancer Center. I've not had the privilege of meeting Dr. Lee, but in my own studies of EMT and bone metastasis, I've certainly become familiar with his research into TGF-beta. So again, thank you for the invitation and congratulations to Dr. Lee. Okay, so the focus of the talk that I'm going to give today is characterizing and using a living biobank platform of advanced prostate cancer to better understand disease mechanisms and to discover and validate treatments. So I'll spend a little bit of time discussing the characterization of organoid phenotypes because people usually are pretty interested in that. And then I will discuss their use in high throughput drug screening. So this slide is probably not necessary for this particular audience, but in case there are a few non-prostate people out there, I just want to introduce the clinical context of the platform that I'll be talking about. So about 15% of primary prostate cancer recurs and progresses to metastatic disease. Androgen deprivation therapy is the universally accepted first-line therapy for progressive disease, which is highly effective but not durable, giving rise to castrate-resistant prostate cancer, or CRPC. More complete AR suppression with antagonists such as enzalutamide and abiraterone have improved survival but have not proven to be curative, and in fact may have helped with the contemporary increases in the extent of AR in different disease that is seen today. So the majority of CRPC remains AR-dependent adenocarcinoma, while about 20% become AR-independent, and a portion of these have the neuroendocrine phenotype. There clearly is a need for new approaches, either alone or in combination with AR targeting. And as I'll show you, and I'm sure you're aware, CRPC is highly heterogeneous, but there have been relatively few unbiased approaches to drug discovery due to the fact there's really a lack of a sufficient number of representative models. So I'm going to talk a little bit about building a platform to do just that. And I'm sure many of you are aware that over the decades, there have been only a handful of prostate cancer cell lines established. And that a platform is needed that represents the underlying genetic mutations and pathology subtypes found in contemporary patient CRPC. So in 2014, Hans Cleavers, Charles Sawyers, and Yu Chen, I think, made a nice advancement in describing organoid conditions to establish cultures from metastatic prostate cancer biopsies. But unfortunately, the community has had a relatively low success rate with establishment of these cultures. So an alternative resource are PDX cohorts, and the University of Washington has built a fairly comprehensive collection of greater than 40 models, mostly from rapid necropsies over the past 20 years. But like patient samples, PDX tumors are quite difficult to grow in vitro. So using PDXs as a source of renewable material, Mike Basheri in my lab has optimized organoid growth conditions in order to improve the efficiency of their establishment. And I'll refer you to Mike's paper for a description of the methods, but I just wanted to take a few minutes for those of you who don't work with organoids to summarize some of the observations. So with respect to LeuCat PDX models, we find that about 90% of them initiate growth as organoids, and about 50% can be passaged multiple generations. They do require continuous 3D conditions, and we have not had much luck adapting them to 2D. The phenotypes and genotypes are maintained, and again, I'll refer you to Mike's paper for data on genotypic stability, and I'll show you a few slides on phenotypic maintenance in organoid cultures. Now, these cultures are useful for mechanistic studies. They can be genetically modified. They can be manipulated for various other types of assays. But I do want to say that in general, organoids grow much more slowly than cell lines. So one has to kind of change your expectations in terms of a timeline. And depending upon what you're doing, we've found that in general, we've had to develop special procedures to work with cultures in 3D. So oftentimes, published protocols have to be modified. With respect to patient-derived organoids from biopsies that we received from the clinicians here at NIH, we find about a 10% establishment rate from soft tissue biopsies. We have not had very much luck with bone metastasis biopsies, and I think that's mainly because there's a very low yield of tumor cells. I will warn you that on average, these cultures require about 6 months to establish and characterize, so patience is needed. But we have found a very interesting range of phenotypes and genotypes, although we haven't been able to identify any clear selective features. So I just want to take a minute and show you how the LuCap platform reflects the mutations in clinical samples. And what's shown on the left here is genomic analysis of 150 metastatic CRPC biopsy samples. This goes back to 2015. There's obviously much more data now, but I think this is very representative. So AR amplification and P10 and P53 loss of function are the most common mutations, and there's a long tail of less frequent but functionally important mutations in clinical samples. And the LuCap cohort mirrors the frequency of the major drivers pretty well, and there are, in addition, several models with rare drivers that make them very useful for mechanistic studies. Now, you might notice that RB loss is a bit overrepresented in the LuCap cohort, which may in itself be a little bit interesting. Recent analysis of consortium data has shown that RB loss is the most predictive genomic change for shortened survival of CRPC. So as I mentioned, I'd like to tell you a little bit about phenotype, because the fidelity of organoid phenotypes is obviously very important to their utility as models. And I want to show you what's a very interesting patient biopsy derived model that Mike Bashiri has been analyzing in detail, and which illustrates how well lineage phenotypes are maintained. So this particular biopsy captured an array of cell lineages that span the adenocarcinoma to neuroendocrine phenotypes. And this is the most heterogeneous model that we work with, and and I think I need to get the pen active here. Yeah, okay, so I can show you what I'm talking about. So what's shown here is single cell RNA seq analysis that depicts a range of lineages. And here is one population that's adenocarcinoma. There's an amphicrine population co-expressing AR and neuroendocrine markers, and there's a neuroendocrine population. And molecular analysis suggests that the lineage differences are epigenetically encoded, which is very interesting and a seminar unto itself, and not really what I'm going to talk about today. But what is particularly remarkable, I think, is that there is a distribution of self-renewing. So on the right here, these are mainly self-renewing cells, and more differentiated quiescent cells. Yet over a 2-year period, the lineage populations have remained pretty stable. So I find this quite remarkable, and I think this data really illustrates that organoid cultures are far less selective for rapid growth than cell lines and can maintain lineage heterogeneity. So in addition to lineage heterogeneity, I just want to show you an example of transcriptionally determined phenotype. And That particular phenotype that I'm going to discuss is the result of interferon signaling, and it is the interferon response signature. So signatures obviously describe phenotype, and it's composed of about 80 interferon stimulated genes, ISGs, which have become of great interest recently because of their potential to influence immunotherapy approaches. So the interferon response signature is observed in tumor cells and can be the result of both autonomous and immune cell interferon. And in the tumor cells, ISGs have been linked to aggressive phenotypes such as chemoresistance. So what is shown here are ISG scores. On the left here are ISG scores that we calculated for patient biopsies from one of the Fred Hutch cohorts. And you can see maybe that in primary cells have relatively low levels of signature, whereas metastatic cells show quite a large range and have population with significantly higher signature. Now, when we look in the LUCAP PDX cohort, we see like metastatic prostate cancer that you have a range of signature expression. And you might also notice, because interferon has become signatures have become very interesting with respect to neuroendocrine cancers, that neuroendocrine prostate cancer models tend to be enriched for the higher levels of for the higher scores. So what's shown here on the right then is a comparison of the relative ISG scores in PDX tumors and the derived organoids. And perhaps you can appreciate that the relative levels are relatively well maintained. We've also used a biochemical marker, which is STAT1 phosphorylation, to validate that the interferon pathway activity, and that correlates very well as also. So I think another interesting point, and the final point I'd like to make here, is that the maintenance of the ISG score in organoids localizes the source of the response to the tumor cells, since organoids don't contain immune cells. So that's always been a confounding factor when you start looking, obviously, at clinical samples. So now let me just return to the work that I introduced at the beginning of the seminar, which is a comprehensive drug screen for CRPC. And just reiterate again that CRPC is genomically heterogeneous, so it really requires a reasonable number of models in order to generalize. And CRPC is dominated by a loss of tumor suppressors, which lack specific biomarker-driven treatment. So it really also calls for a non-biased approach. We are starting by identifying individually active drugs in the anticipation that combinations will be needed. So this just shows how the screen is done. The screen was done in collaboration with the National Center for Advancing Translation, or NCATS. And the way the screen is done then is to harvest PDX tumors into single cells, which are then plated in Matrigel into a 384 well format. Importantly, we use a 10 concentration dose, I think, which ends up giving us very reliable data. The cultures are drugged on day 3 and 7. I think I've got something in the way here. Sorry, I was close. Day 2 and 5, and terminated via cell titer glow on day 8. The data is then processed and using a variety of controls to allow normalization and comparison of runs, and then is analyzed and visualized using a Palantir, using a platform that Palantir developed for NCATs. Now, this method was developed and validated, and most of the data was collected by a postdoc in my lab, Keith Jansen, who has since gone on to work at GSK. So in terms of what we have actually screened, the screen includes data on 126 compounds. We have looked at a total of 32 unique models, including 4 PDOs, 14 adenocarcinomas, 6 neuroendocrine models. Because many of the adeno models are sensitive to castration of the mouse host, we have also looked at 5 experimentally selected castrate-resistant adenocarcinomas, 2 amphicrine models, 1 double negative. We've looked at LNCAP and RWPE in both 2D and 3D, and we have also looked at normal prostate basal cells. We do remove curves that don't meet quality control. That's been a relatively small percentage, about 10% of the screen. And importantly, we've also done total transcriptomic sequencing of G1 organoids that were derived very similar to those cells that were used in the screen. So, okay. Oh, okay. I don't know why it wouldn't advance there, but I'm glad that it did. So this is just a description of the drugs in the library, and we've tried to focus on designing a highly actionable library of compounds. We have looked at 80 compounds, have been conserved from beginning to end. We did have 26 drugs that were removed halfway through the screen because there was just simply no response. And we added at that point 20 new compounds that had for some reason come to our attention. So you can see here the categories of drugs that we've looked at. The most common category is drugs that target cell signaling. 21 of the drugs that we've looked at are FDA approved, 45 are in clinical trials, and 34 are still in preclinical states. These include almost all of the epigenetic compounds. Now, defining responsiveness is really fairly qualitative and will vary obviously somewhat from drug to drug, but a pretty reasonable response criteria that I'm going to use here for translation into the reasonable for translation into the clinic is an AC50 of less than 1 micromolar and a maximum kill or response of at least 80% at the highest drug concentration. So this is just a snapshot of the drug response distribution using that definition of response and non-response. So we're really looking at black and white here. And this shows the response for 80 drugs that were assayed in every model. And as I said, there's some additional drugs that were assayed in most, but not all, and they're not included here. And so what you see then in the rows are the various different models that have been organized relative to their lineage definition, and in the columns are drugs that have been organized relative to their classification. And maybe you can appreciate in this responder, non-responder type of showing that there is a wide distribution, there's a very wide distribution of responsiveness from drugs that inhibit every model to drugs that inhibit none. We think that the most interesting drugs are those that are in the middle of the range because response by some models and not others, of course, suggests some specificity to the efficacy. Now, maybe you can appreciate that the drugs targeting cell signaling are a real checkerboard, but there are also classes of drugs that show lineage clustering, and I'm going to return to a more in-depth analysis of those. So just a couple of slides of very general conclusions before we move on to more in-depth analysis. I just want to emphasize that drug sensitivity is not determined by growth rate. That probably is related to the 8-day response window that we're looking at. This isn't necessarily true of cell line screens that are usually done with a 48-hour window. Oftentimes, growth rate is a confounding factor. So I just want to mention that when we looked at LNCAP in 3D and 2D, we see mostly overlapping drug responses, although there is some increased sensitivity in 3D to a few drugs. What was extremely surprising to me is that there were 76 compounds that had no responders, and this included drugs that have been published as having activity in prostate cancer cell lines. So I think when you see a drug with no activity across multiple models, this obviously has to lower the priority among competing clinical trials. And there are 50 compounds that had at least one responder. So a few other general considerations. So maybe a little bit surprising, there was no statistically significant differences between the castrate-resistant and parent adeno models in terms of drug responses. They were just qualitatively actually identical. So no gain sensitivities or loss sensitivities. Also a little bit surprising to me, compounds targeting cell metabolisms were pretty much generally inactive as single agents. That may relate back to the relatively rich conditions that are needed for organoids, but I think it is something to keep in mind. The neuroendocrine and adeno models were pretty equally responsive, although maybe the neuroendocrine models had a slight edge. And then building upon the screen data, we've done a number of experiments with various organoid models to dissect the parameters of responsiveness. And that's really what I'm going to talk about now. Before I do that, though, I just want to really quickly address the issue of reproducibility. It's something that I'm asked about a lot. So I just want to tell you that in addition to doing biological replicates in the screen for many of the models, we've also used an independent method to validate the drugs, and that's essentially testing them by hand at the bench. So what you see here is on the left is normalized AUC, which takes into account the dynamic range of response. It looks at basically the area under the curve. And so what you can see when we look at normalized AUC in the screen versus repeats that we've done by hand, we see a very high correlation value of 81%. So this is really made us confident that our data is robust enough to be considered as a continuous variable, which I'm going to now use in a variety of different analyses. So an important question is obviously the transcriptomic and genomic correlates of response. And Ilya Senatorov has done these type of analysis that I'm going to talk about here and in a few subsequent slides. So what is shown on the left then is the normalized AUC to selected compounds, and we're comparing RB mutants and non-mutants. So as you can imagine, RB mutants are enriched for neuroendocrine models, but we also include a number of adenocarcinoma models. And what you can probably appreciate is that there's an interesting correlation between RB loss and responsiveness to inhibition of anti-apoptotic BCL2 family members, as well as correlations with responsiveness to chemotherapeutics and checkpoint inhibitors. So when we actually do a statistical analysis of the average AUC for RB mutant versus RB wild type models, what we see is that there are a number of drugs that show a statistically significant difference. Now, somewhat interestingly, this did not hold true for P53 mutations. And I want to take just a couple of minutes to discuss the specificity of targeting the apoptosis pathway. So for RB mutants, I just want to emphasize that targeting BCL-XL with either navitoclax or the more specific compound WEHI 539 is efficacious, but targeting BCL2 with venetoclax has no activity. Now, targeting MCL1 can be efficacious, but it is not RB dependent in its responsiveness. And then, as I mentioned, there are other compounds that correlate, whose responsiveness correlates to RB mutation, and I'll come back to those in a minute. But I just want to also say that the drug specificity for inhibition of anti-apoptotic proteins is not correlated with levels of expression. So you can see on the left our transcriptomic levels, and the neuroendocrine models have asterisks, and on the right are Western blots, so protein levels. And BCL2 is much higher in neuroendocrine prostate cancer, whereas BCL-XL and MCL1 have pretty uniform levels, even though they, and not BCL2, are the targets of differential sensitivity. So I think this is actually fairly consistent with a number of other solid tumors where BCL2 doesn't seem to be an important determinant of anti-apoptosis targeting. And I think it really shows us that to interpret the specificity, we need to understand the networks regulating apoptosis much better. So to begin looking at patterns of resistance and sensitivity, Ilya has analyzed the range of responsiveness on an individual drug basis across multiple models. So what's shown here on the left are the response of all of these models to docetaxel, looking at the standard deviations from the mean. And he's done this for all of the different drugs. And basically this is, what you see here is then a distribution of the variants, and here is just a tabulation. And so using this data, docetaxel was the drug with the highest range of responses. And the screen, he then ranked the screen based on docetaxel response. and using the top quartile of response and the bottom quartile of non-responders, used ANOVA to check whether other compounds are enriched in terms of sensitivity to strong versus weak responders. So this is a way of looking for drugs with similar patterns that may be synergistic in their activity, or looking for drugs with anti-correlated patterns, which may be useful in overcoming resistance. And so this is just a graphical depiction of the docetaxel ranking here relative to the models and a number of different drugs, and just shows the similarity of distribution of sensitivity. So then doing the statistical analysis, what is shown here in this particular table is that various chemotherapeutics, anti-apoptotic inhibitors, and checkpoint inhibitors are statistically associated with docetaxel responsiveness. And so this includes navitoclax, for example, that we've talked about, and drugs that target the G2M transition. So this then is just a model of the fact that the DNA damage response and apoptosis pathway form an interrelated vulnerability network. So sensitivity to DNA damage, we know, is increased by DNA damage response mutations and replication stress, which is probably very relevant for RB loss. The therapeutic index for chemotherapeutic agents is really based on the fact that aggressive cancers have more trouble repairing their DNA than normal cells. And because cancer cells have lost cell cycle control, particularly in the G1S transition, they're more dependent upon checkpoint control to gain sufficient time to repair their DNA. So that's the left side of this model. On the apoptosis side of the equation, we know that death resulting from DNA damage requires an intact apoptosis pathway. So if the BCL2 family members that execute apoptosis, so back and back, are deleted in experimental models, those models lose sensitivity to chemotherapeutic agent and other DNA damaging agents. Now, the observation that CRPC inhibition of anti-apoptotic family is sufficient to activate death, at least in vitro, suggests that there's a high level of constitutive stress in these cells, that they're basically on a precipice and they just need a little shove to undergo the apoptotic execution. So I think the point we are at here is that our translational objective is to find the most efficacious and least toxic combinations that take advantage of these vulnerabilities. And then I just want to touch very briefly on the fact that with sufficient models and transcriptomic analysis, it's possible to analyze for the pathways that enrich for responsiveness or resistance. And I know this looks like a hot mess, but the yellow circles represent pathways and the red circles represent different nodes in these pathways. And so just to deconstruct this a little bit, I've listed here a few interesting pathways that distinguish responders from non-responders. So non-canonical WIMP, for example, interferon, as we perhaps would have predicted. As we had obviously hoped and expected, apoptosis is a difference, as are ER stress, protein folding pathways, and there are also some very interesting differences in metabolic enzymes. So to really show how we can use this, what's shown on the right then is a more granular analysis of the death receptor pathway within that big apoptosis pathway analysis. And what this shows, for example, that the apoptosis inhibitor BERK3 is much more highly expressed in non-responders, shown here in gray, than responders, shown here in gold. So just to summarize it, this is obviously very early days, and I'm showing you a quite superficial analysis. But I think you can see how having sufficient number of models with robust quantitative response data can really assist in looking at mechanism. So now the elephant in the room, the $64 question, is how predictive are in vitro responses relative to in vivo responses? And we're tackling this question in collaboration with Eva Corey. Unfortunately, COVID has really slowed the preclinical trials that were planned. So we really need to gather much more data. But I would say so far there are not perfect correlations, and we are probably going to see some drugs correlate much better than others. But for a limited amount of data that we have, I think the trends are promising. Now, these PDX preclinical trials are obvious with subQ tumors are really the accepted standard for in vivo responses. But when we think about CRPC, the clinical context that could be very useful is early recurrence following androgen deprivation. And the most common site for reoccurrence is the bone. So Ivy Yin in my lab has developed models, I think, that will be very useful for analyzing drugs in this context. And I just want to show you a little bit about those. So Ivy has developed 3 highly reproducible AR-positive bone metastasis models from LuCap PDXs that really represent the heterogeneity of bone metastasis histologies. And maybe some of you know that it has been very hard to get AR-positive bone metastasis models. Pretty much the ones that are available today are AR-negative cell lines. And so what's shown here are 3 of the models, one of which is so you can see the histology here of tumor growing in the bone, AR positive. It turns out that these mets are osteoclastic. The model is extremely efficient. It's basically 100% efficiency. So let me back up a minute. I should have mentioned that the way these models are made is to use intracardiac injection of organoids that have also been labeled with bioluminescent marker. So you can see in these various models that the metastases localize mainly to the vertebrae. Sometimes there are mets in the skull. Sometimes in the long bones, there is a little bit of disease in soft tissue. It depends somewhat on the model, but it's minor compared to the tumor burden that's found in the bone. So this LuCap 136, as I said, is osteoclastic. LuCap 167 actually gives a mixed model of both osteoclastic and osteoblastic metastases. It's also relatively efficient, so easy to use. LuCap 23.1 gives a beautiful osteoblastic model It's actually a little bit more difficult to use because the Mets grow much more slowly and the efficiency of establishment is around a third of the mice. But I think these are, these new models have a number of potential utilities. And so I just want to talk a little bit about mimicking clinical disease then, and using the LUCAP 136 model. So, and showing you an example of how the model responds to castration response. So the mice are castrated once bone metastasis is confirmed, which inhibits growth in the bone for a few months. And you can see that here from the BLI images. It's been graphed here. But eventually the bone metastases begin regrowing. And the mice, which takes a couple of months, and the mice actually become paraplegic, and most of them have to be sacrificed for that reason because they become dehydrated and cachexic. So this is obviously so this is a model where we can really look at the very interesting question of what the biology of recurrence after castration looks like in these various models. And here again, comparing intact Mets to Mets after castration, and of course we can look both early and late. So what we see is that compared to an intact mouse, there's been the tumor has regressed significantly. In the intact mouse, again, the Mets are AR positive. It's a little bit hard to see here. There are some cells that are weakly positive for AR in the cytoplasm, but clearly there is not active AR signaling going on. And we have also used BRDU labeling, so the mice are injected with BRDU an hour before they're sacrificed, which really allows us to look at the proliferating cells. You can see in the intact mouse there are a lot of proliferating cells. It's about 15%. Whereas in the early castrated mouse, their proliferating cells are present, but they're there in relatively low numbers. Now, in terms of looking at the biology of what's going on, we have begun by doing RNA-seq on the harvested metastases. And I'll just show you here is shown differential gene expression in castrated versus intact models, either harvested early after castration or late. And probably one of the main things that you can see is that the surviving cells actually are starting to express what we recognize as neuroendocrine markers. So this is just a really quick tour of these models. We're really pretty excited about being able to use them to look at both bone metastasis biology as well as using them as models for drug treatment. So I am going to stop here and just conclude by saying that I think as a community, if we're willing to do the hard work of taking principles and concepts from basic research and testing the generality across multiple patient-derived models, we'll move the fruits of basic research to the clinic much more efficiently. That's my bottom line. So let me just again acknowledge the many talented people that have been involved. Hopefully, I pointed out the postdocs that have done the work: Ilya, Keith, Mike, Supreet, and Ivy. Brian Capaldo is our bioinformatician, and sort of everything revolves around him, basically. I would also like to thank Adam Sawalski, who is a tenure track investigator in the department. He's helped us tremendously with genomic analyses. And to thank Fatima, Bill, and James, who work with us to get us clinical samples and discuss their significance with us. And of course, most of this work has been a collaboration with Eva Corey at the University of Washington, as well as Pete Nelson and Ilse Coleman, who have supplied a lot of molecular data for the LUCAPs. And I will stop there and take any questions.
Thank you very much, Kathy. So if there are questions, I think, Akash, you want to go ahead with your question?
Sorry, I just put it on the chat function. Hi, Kathy.
No problem.
Hi, Akash.
Great talk. Thanks for sharing all of your exciting data. I was just wondering about whether you've looked at generating humanized organoids with reconstituted, human immune systems like CD34 positive hematopoietic stem cells to study the impact of therapeutics in an immunocompetent context, because obviously that would be very important from a translational standpoint.
Yeah, no, that is a great point, and I completely agree with you. We haven't tried any of that yet. We're still thinking about it, and everything got so slowed down with COVID, but I think it's a great point, and I think you're absolutely right.
Kathy, I have a question, and this may be a crazy idea. In terms of the organoids and in terms of looking at the effect of the environment versus the tumor, has anybody done, like, taken a sample from a human tissue, whatever it is, and then put it in different parts of an animal, a mouse, let's say, and see what over time growth and then molecular profile and differences based on the regions? Like, you pick up, say, a met from a lymph node, you implant it in a bone of a mouse, then put it in a lymph node. Maybe this is a crazy thing, and just look at the effect of the environment on the actual tumor.
Yeah, no, it's not crazy. It's a good point. And I wish I could recall some of the data better. So definitely that has been done in the metastasis field. I don't think anybody's done it with prostate that I know of, but environment definitely matters. Exactly what the effects are, I think, are going to be extremely context dependent. We are trying to compare the LUCAP models in the bone versus subQ tumors versus implanting in the prostate. And we'll see whether castration resistance is changed in any way by that, or what the molecular effects are. So it's a good point. I wish I knew some of the other metastasis literature a little bit more thoroughly, and I could probably give you a more exact answer.
No
but
problem.
It's a good thought.
Thank you. Russell, you want to
Sure.
ask your question?
Hi, thank you for the great talk. I was sort of intrigued by your definition of response and, how much that skews your readouts for drug activity based on the assay, the cell titer glow, and so forth. And in particular, hormonal therapies, drugs like enzalutamide that, we use at 10 micromolar. I couldn't even find in the literature search an IC50 for the drug. So what other assays are you looking at to get at some other different phenotypes? So that's two sort of parts of the same question.
What other assays are we using? So it's a good point, and I understand what you're saying. So people will use apoptosis related assays, for example, or, different metabolic indicators. For us, it's really kind of a bandwidth and a practical issue. So setting up and doing high throughput screening, you're just kind of limited in how much you can do. We're limited by the infrastructure, we're limited by cost. That doesn't mean that you couldn't, that we couldn't take the results from the high throughput screen and look at other variables using different assays. So, I'm doing a heck of a lot of hand waving here because we haven't really spent a lot of time looking at other assays, but there are other assays available, and it definitely is a good point. I guess that we're pretty much at a point in the analysis of the data that hasn't really come up as a significant point yet, but it certainly will.
And sort of on that level, are hormonal modulating drugs, are they performing the way you would expect them to? Are drugs like apalutamide, enzalutamide that have gone through to FDA approval and we know have clinical benefit, do they reproduce?
They reproduce, but you need to use so much in vitro in these systems that you kind of we're right on the edge of non-specific effects. So we get partial responses to AR inhibitors in vitro. So we get nice, strong responses in vivo, but in vitro, the responses are not nearly as robust. So that makes it difficult to combine androgen receptor antagonists or inhibitors with drugs directly.
There's one more question from Tim Lane. Tim, you want to ask your question verbally?
Kathy, that was a great talk. Very few people bring up cachexia. I thought it was very interesting that you mentioned that this last model, this AR positive model, this was a important endpoint for you. And I just wanted to ask, because I don't know about the prostate mouse models with that endpoint, how unique is this? And are you planning on looking at cachexia specifically as a point of modulation?
Well, I don't know how directly cachexia is actually related to the model itself, to the physiology, to the biology of the model, because it's confounded by the fact that once the mice get paraplegic, it's very hard to feed them and keep them hydrated. That being said, cachexia for metastasis models is a very common endpoint in the mouse. And we will probably, for the time being, just use it as a survival endpoint. But obviously very, very, very important for in considering patient care.
Okay. We have a couple of more questions. Debu, you want to go ahead and ask your question?
Sure. Thanks for your talk. So you did mention about the epigenetic modulators, and I was wondering what's the nature of those epigenetic modifiers? And secondly, when you treat the cells with those compounds, does it decrease the cellular or tumor heterogeneity to allow for other drugs to be more effective?
No, that's a great point. I can't really, so in terms of the epigenetic modulation of that particular model, it is driven by ARID1A, ARID1B mutations in the model. we have not found epigenetic modulators to be particularly effective in the model, and we have not found drugs that really differentially select for one lineage over another, which is kind of surprising, but those are the results.
And I think we have one last question from Priyanka. You want to ask the question, Priyanka?
Sure. Thanks for a great talk. I just have one quick question about your organoid models. So what is your correlation in terms of maybe gene expression or maybe other sequencing data sets that you possibly have, all the way from the primary that you're preserving down through your PDX and organoids? Because when you look at PDXs, there's definitely differences in terms of how fast they engraft, and converting those into organoids obviously do take a long time to grow out in culture. So how much of that are you actually preserving across the samples?
I'm sorry, I don't, you mean relative to the
Primary sample.
To the primary sample? So that's really hard for me to address with respect to the loop caps, since that data is really in the hands of the University of Washington. But I can tell you with respect to our patient-derived organoids, we get really good matching between the genomic analysis that's done on the biopsy and the organoids. So we don't, there doesn't seem to be a strong selection.
Thank you.
One more question from Anthony. Anthony, you want to ask your question?
Hi, yes, Kathy, thank you so much for the talk. This is super interesting. You know, you spoke earlier about kind of the paucity in cell line models being developed from prostate cancer and from prostate cancer patients. I'm wondering, of your PDOs, how many have been derived from patients of West African ancestry or African American patients with prostate cancer, as we start to think about, you know, beyond kind of the socioeconomics and other factors involved in cancer health disparities? It seems like that this could be a potentially a great model for trying to understand the biology, if it exists, of prostate cancer disparities. So I'm just wondering.
Yeah, I don't think any of our models actually came from African Americans, but I absolutely agree with you. Even though the efficiency is low, I would love to see the community work to get a cohort of African American organoids, for sure. You know, since the efficiency is low and getting metastatic biopsies is not particularly easy, it really probably will require a community effort. Yeah, but I agree with you. Be great.
Well, thank you very much. Are there any final questions? Kathy, thank you so much for joining us. It was really a fantastic talk, and we really enjoyed the conversation. And we actually look forward to even potential collaboration.
That would be great.
Yeah, absolutely. I guess I'm going to clap and welcome you. And Dr. Lee, do you want to say something? You're muted. You want to unmute?
Okay, can you hear me?
Yes.
Yeah.
Okay, I just want to thank Kathy for taking time speaking to us, and we really appreciate it. And that's about all. I'll talk to you later. Thank you.
Thank you so much. It's nice meeting you.
Thank you, and hope maybe next time it'll be in person.
Yeah, that would be great. I'd love to come.
Yeah, exactly. Be well and be safe, and thank you very much.
You too.
COVID-19 vaccine is available. We can come to Chicago again.
That would be great.
Yes, or go anywhere in the world. Absolutely.
Thanks so much. I really appreciate the invitation.
Thank you.
Take care, everyone.
Thank you very much.
Bye-bye.
Bye.
Bye.